Hey now, I'm Jack Cush, executive editor of RheumNow.com. This is the 26th of May, 2017, and you're listening to the RheumNow Week in Review. Highlights in the news this week is IL-6 is a hot topic, especially as far as new regulatory approvals. Additionally, a major new osteoporosis drug in development may have hit the skids for heart reasons. More on those later. At the top of the news, an interesting study done with 24 patients with erosive RA looked to see if whether you could use an anabolic steroid, teriparatide, to prevent the development of erosions. In this one-year study headed by Dan Solomon and Ellen Gravelis, they actually did not show that the use of the anabolic steroid provided any reduction in either the rate or the volume of erosions that were seen in the 24 patients that were treated. There have been some studies in the past that looked at bisphosphonates as potential add-ons to reduce, or Prolia for that matter, again with some variable results. While it makes some sense to make this approach in an erosive disease, it seems better to control the disease with inflammation, and that's where anti-TNF inhibitors have been particularly effective. Interesting study comes out of Sweden, a large cohort study, a nationwide study of patients with ankylosing spondylitis, psoriatic arthritis, and undifferentiated spondyloarthropathy. Looked at over 27,000 patients, compared them almost 5-to-1 to a normal control population, and looked at the rate of acute coronary syndrome and strokes. And in fact, they showed higher rates of acute coronary syndrome and stroke in patients who had these inflammatory disorders. Not surprising, as we know that inflammation does drive cardiovascular risk in rheumatoid arthritis and some other disorders. And this has been seen before, but what's good about this study is it comes from a very reputable group, very large cohort study, very well done research. Again, underscores the importance of controlling inflammation to control the untoward effects of inflammation. A nice study looked at the potential biomarker for IL-17 driven psoriasis. We know interleukin-17 is very important in the pathogenesis of psoriasis. That what drives that, however, is not known. IL-17 is very much involved in defense mechanisms and mucosal immunity and whatnot. And in this particular study, they looked at 8 patients with and without psoriasis, and they analyzed 170 or more proteins from the skin, both lesional and non-lesional skin, and from blood, and to see what might correlate with IL-17 or markers of a disease activity. And they found that beta defensin 2 was really quite a good biomarker. Number one, it's fairly easy to measure and seemed to have a high correlation with PASI skin scores and also in IL-17A levels. So again, this represents a potentially useful biomarker for the future. The TICOPA study is a tight control in psoriasis and psoriatic arthritis trial that showed that, as it did in RA, the tight control and aggressive therapy had certain benefits. However, a new financial analysis looked at the cost effectiveness of tight control. Specifically, they looked at quality adjusted life years, and showed that tight control was more costly, almost twice as much more costly, and also had a significant increase in qualities, but was not necessarily cost effective in most analyses that they ran. What they did see, however, was that certain subsets of aggressive patients might make some sense to use a more aggressive regimen, as was done in TICOPA. So sort of bad news that sometimes using more expensive therapies isn't always the best or most cost-effective measure. A very interesting study looked at the infection rate in children with JIA who go on biologics. And specifically, they looked at serious bacterial infections. In a study of 2,495 JIA patients, they showed, and this is claims data, that there was a much higher rate of serious bacterial infections in those who are taking TNF inhibitors versus those who are just taking DMARDs. Now, there could be some selection bias here. Obviously, sicker patients are going to get a TNF inhibitor. But nonetheless, it is something that's usually not been seen in other studies, that, again, it is a significant increase in SIEs, and that, it's something that it might be a true message, especially in children who have just pure inflammatory disease and haven't been on a lot of other therapies. So a little bit of warning there. A study of patients who have ANCA-associated vasculitis, 323 patients, and compared what the subsequent cancer rate might be if they were treated with Cytoxan or Rituxan. And we know from other studies that such patients may be at higher rate of certain cancers, especially if they receive cyclophosphamide, where there's a high rate of urogenital cancers, bladder cancers. And in this particular study, they showed that the cancer rate was higher with an SIR of a standardized incidence ratio of 3.1 for Cytoxan, and that was 4.5-fold higher rates of cancer compared to Rituxan. So the significant risk with Cytoxan, 3.1 SIR versus 0.67 SIR with Rituxan. In Rituxan, they found that the cancer rate was roughly equal to the population rate. So obviously there's a significant risk to using Cytoxan or cyclophosphamide in patients with ANCA-associated vasculitis. The two big bits of news from the regulatory front this week from the FDA. On the same day, the FDA approved the use of Kevzara, the generic name being sarilumab, an IL-6 inhibitor, and this now becomes the second FDA-approved IL-6 inhibitor for use in patients with active rheumatoid arthritis. Kevzara has been approved in Canada and other countries, and the interesting thing about this drug that's being developed by Regeneron and Sanofi is that it comes into the market priced 30% below the current TNF inhibitor price, which is roughly around $49,000 and $50,000. This comes in at $39,000. So aggressive pricing for a brand new product. IL-6 inhibitors are going to expand. We now have obviously Actemra and now Kevzara in the market, and there's another one soon to follow. Speaking of Actemra or tocilizumab, the FDA approved this week Actemra for use in patients who have giant cell arteritis. Really based on the GIACTA study, this drug was sort of given a fast track to FDA approval, was approved as a subcutaneous administered drug, and it says in the package insert that it should be given along with tapering doses of corticosteroids. So the idea of giving this drug so that you can get patients off of their corticosteroids, as demonstrated in the trial, is also demonstrated or reflected in the package insert. The ACR has actually been very active. If you haven't noticed, under Sherrod Lockenpal, they've had a lot of announcements about position statements, responses to what's going on with Trump Care and whatnot. The ACR issued another statement this week supporting the FDA's draft on interchangeability. They support the FDA's position on naming suffixes, meaning that infliximab is not just infliximab, it's also called infliximab DYYB for the new one, Inflectra, et cetera, so that we can distinguish between the generic infliximab and the biosimilar infliximab. And they also support the Biosimilar User Fee Act, which the FDA is considering. But they actually had a strange, but not expanded upon statement about extrapolation. They did not or question the issue of extrapolation. Extrapolation being that if a drug is studied and studied in rheumatoid arthritis and gets approved as a biosimilar, it gets all the indications of the biosimilar or the originator it was being compared to. So you could do just a study in psoriasis, yet get an indication for rheumatoid arthritis, JIA, Crohn's disease, and ulcerative colitis, even though you didn't do those trials. That's extrapolation. And you may not like it, but that's the rule. And the rule is really based on if you prove biosimilarity, if you prove structural, chemical, functional, and mechanism of action, biosimilarity through analytical methods, then all you need is one clinical trial to get the drug approved, and then you get all the indications. So it's not something that's really up for debate. And on why they didn't approve it or have a problem with it, worldwide, no one else is having a problem with it. A survey in the UK of 364 patients looked at patient attitudes about how they receive information on TNF inhibitors and showed that a third of patients who got information about the new use of a biologic, a TNF inhibitor, said that openly it helps them, and another third said openly that it hurts them, meaning it scares them. So, where's the fine line? There are many who have sort of misconstrued impressions about biosimilars, about biologics, and their safety, especially their safety. But it is the idea that giving patients more information may, in fact, be a deterrent to the use of the drug, filling the prescription, and being adherent. My overall rules is give the patient written material to tell them to read, question, circle, and come back, and not to make decisions without talking to me. But I more specifically focus on 3 things that they can expect that are most common side effects. I also give them the 1 or 2 or 3 things that are the most dangerous that might avert their use and explain why that's a 1 in 1,000 risk, and that's a low risk compared to the risk of the disease that they're trying to treat. Last week I was at a state society meeting in Puerto Rico, and Dr. Doric Erkan was there talking about antiphospholipid syndrome. He's an expert from Hospital for Special Surgery, and I came away with 2 pearls. One, that in treating secondary thrombosis prevention, the current management of those venous thromboses would be to use warfarin and keep the INR between 2 and 3 for the venous thromboses, but that if you're treating arterial thromboses, that you really want to keep the INR between, 2 and 3, but also use aspirin. If you're not going to use aspirin and you're dealing with an arterial thrombosis, you probably should keep the INR between 3 and 4. So again, that's venous thromboses. It's without aspirin, it's 2 and 3 is the INR with Coumadin. But if it's arterial thromboses, use aspirin, and same INR of 2 to 3 is suggested. But if you're not going to use aspirin, can't use aspirin, you need a higher INR, 3 to 4. He also came across with the management of catastrophic antiphospholipid syndrome currently is anticoagulation, high-dose steroids, IVIG, often with plasma exchange, and there's some suggestions some patients might even benefit from the use of rituximab. An important development in the news came this week with the announcement by Amgen and UCB about their drug romosozumab, which is also called Evenity, Evenity. This is an anti-sclerostin drug that has a great deal of promise in the treatment of osteoporosis and strengthening bones, and they've had a number of phase 2, phase 3 trials that look very good, highly promising in their ability to reduce fracture rates overall. But they announced the results of the ARCH trial, and what was unique about this trial was, one, its design. It was romosozumab given for 12 months, followed by alendronate, and then a matched placebo control who didn't get romosozumab, who took alendronate. And they showed there was a significant reduction in both vertebral and non-vertebral fractures at the end of 24 months. The problem was at the end they also noticed an imbalance in cardiac events, the nature of which was not disclosed. It was reported that 2.5% of those on romosozumab had these cardiac events versus only 1.9 in the alendronate only group, suggesting this could be a potential problem. So there's a lot of buzz about this. This could lead to a stall in the drug's development or its approval, or could lead to an FDA hearing. Still a lot of unknowns, but a drug that was previously thought to be fast-tracked for approval and success now has a wrinkle or a bump in the road, let's say, in its development. And lastly, there was an important report from the New England Journal on the use of an anti-IL-5 monoclonal antibody called mepolizumab. And it was used in patients who have Churg-Strauss vasculitis, now called eosinophilic granulomatosis with polyangiitis, or EGPA. In this particular study, reported in New England Journal, 136 patients with relapsing or recurrent disease were given either placebo or 300 milligrams of mepolizumab every 4 weeks. And in the end, that the patients who received the anti-IL-5 monoclonal antibody had a higher percentage of remissions at 48 weeks, 32% versus 3%. And the number of patients who did not achieve remission was much lower on mepolizumab, 47% versus 81%. The interesting thing about this is when this drug worked, it worked on the components that might be IL-5 driven, meaning the asthma symptoms and the sinusitis nasal symptoms, not as much on the vasculitis symptoms. So this drug needs to be studied with maybe in combination with other drugs that are better at vasculitis. And it's unclear, although it's very promising, it's unclear exactly who should get this drug going forward in the future. And one more, lastly, I have actually a report about Still's disease, my favorite disorder. This is a study of 34 patients with adult onset Still's disease and looked at the association of Still's disease with the NLRP3 gene that is important in activation of the inflammasome, which drives the systemic inflammatory disorder. While everyone assumes that Still's disease is an autoinflammatory syndrome based on its responsiveness to IL-1 inhibition and the symptomatology, which is very akin to that seen with all the other autoinflammatory syndromes, the direct connection with activation of the NLRP3 gene and the inflammasome hasn't been well demonstrated. So in this particular study, they looked at 34 patients and controls, and they showed that there was significantly higher degree of mRNA expression of the NLRP3 gene compared to controls, and much higher. Moreover, NLRP3 mRNA levels correlated highly with Still's disease activity measures. They did further experiments that looked at what happens if you activate the gene with a TLR7 ligand, and they showed that if you can activate NLRP3 in these patients in their PBMs, that you actually have even higher levels of NLRP3 expression, and if you can inhibit it, you have even lower levels, also done in the same patients. All of which is to say it's a fairly good connection here that says that the NLRP3 gene is activated, turned on, and is probably driving patients with Still's disease. We do not know what the instigators are of that process, and it's obviously not genetically based, as are many of the other autoinflammatory syndromes, which are monogenic, often appearing early life. This is an acquired or a developmental defect that occurs that leads to this periodic fever called adult onset Still's disease. That's it for this week at RheumNow.com. Go to the website to see all these citations and more. Tune in next week for more news from RheumNow.com.